Welcome to Biology 2402 Lecture Series, Chapter 28, The Reproductive System. In this chapter, we're going to see the male and the female reproductive system and how it relates to some of the other chapters. Now, the chapter that it'll most likely relate to is the urinary system. If you recall, in Chapter 26 on urinary, we had pictures that went with both the male and the female reproductive. So we're going to have that again. Now, starting with our first slide, we have this picture. Now it says, the reproductive system is the only system that is not essential to the life of the individual, but it does affect other systems. So if we have a urinary issue, it can be a reproductive issue, or a reproductive issue can actually be a urinary issue as well. So it does affect other systems. Now, one of the things that we're going to learn is that the reproductive system is highly controlled by the neurological and the hormonal. So again, Nemesh's 95% rule on electrical and chemical. Next slide. Here we see a slide that you should know by now. So this is something basic that you should have learned prior to, and that is the word gonad that you see there. Gonads are organs that produce gametes. Now, gametes are the sperm as well as the egg. So the gametes are sperm as well as the egg. Notice again we see words like ducts. So ducts will receive and transport the gametes. Then we have accessory gland, which secrete fluids again into ducts. Anytime you see ducts, you know that we're dealing with the exocrine gland. And then we have an area called the perineal area, often called perineum. And that is the triangular shape that we'll see later between the legs. And that's the best way to remember it. So far we have these two slides. Next slide. Here we have the reproductive tract, male and female. Now the terms that you see here, the information that you see here, are general information that you should know by now, such as male and female are functionally different. That sort of makes sense. Female produces one gamete per month, and it retains and nourishes the zygote, whereas males produce large quantities of gametes, the sperm, such as 500 million sperm per day. So this slide is a generality that you should keep in mind. Next slide. Again, there we see our male reproductive now, the testes or the male gonads. The testes deal with the male sex hormones, the androgens like testosterone. And of course they produce male gametes, which we now call sperm. Next slide. Now here's the slide on female. The females have the ovaries or the female gonads, and they release one immature oocyte per month. Now of course that statement is a generality. This is how we end up with having twins, where we have more than one oocyte removed or released. So there are variations that we need to keep in mind. Then we have the ureter tube, also commonly called the fallopian tube. So the ureter tube or fallopian tube. And within that ureter tube or fallopian tube, that's where we're going to see fertilization occur. So fertilization actually occurs in a portion of the fallopian or ureter tube. Then we see the uterus, mentioned at the bottom there. And keep in mind that the uterus has linings very similar to the heart, in the sense that we have what's called myometrium, which is a heavy muscle layered structure. And it's that heavy muscle layer that's going to play a role in the uterine contractions later. Then we have the vagina at the bottom there. Now, one of the things you need to understand is that the female reproductive is far more complex than the male. The reason why that's important to understand or appreciate is that the female reproductive is the one that's going to lead into the developmental chapter of chapter 29. And not only is it a textbook issue, but it's also a nature issue in the sense that it involves more of the female story. So if there's something that goes wrong, chances are, if it's not a problem with the sperm, the chances are more of the issues will arise with the female. So it's a statement I want to make up front and make clear with, because it is often a female issue if there's no issue with the sperm. Next slide. We have the spermatozoa, and this is the pathway to keep in mind. It is a very simplified pathway. Now, you know all right, if you watched any of my previous videos, that if I have a slide that has only terms or places like this, where are you going to need to see them? On the model, for the lab day, for the lab exam. So your object of the game is when you see a slide like this, you need to actually be able to apply it to the model on lab day. So the application of the notes will be important. Next slide. Then we have accessory organs. These two, again, you see simply listed here. Well, list is good, but you actually need to know what comes out of each and where these are located. So the location and what they do. And isn't that the title of our course, Anatomy and Physiology? So the anatomy being the location, the physiology being the function. Next slide. Okay, we have the external genitalia of the male, which is the scrotum and the penis. And of course, you know that the scrotum houses the testes. And then the penis having a portion of the urethra within it. So there's really three pieces of the urethra that we'll see later. Next slide. This slide is now the one that deals with the male. And in this slide is very similar to the picture you saw in the urinary chapter. So this slide is important for the lab exam day. Now you can see that we have different pieces of the urethra all labeled for you. So we have a prostatic urethra. There is a piece not labeled right about here called the membranous urethra. And then we have this piece right here called the spongy urethra. So two of the three are labeled for you. The prostatic urethra, and right about here would be the membranous urethra, and then the spongy urethra. Now, before I go to the next picture, I want to briefly go over the slide that you saw before about the sperm's pathway. So the sperm are produced within the testes. So the sperm are produced within the testes. And then they are transferred to the epididymis. Now the epididymis not only stores the sperm, but also helps mature the sperm. So maturation and storage is a function of the epididymis. At the time of ejaculation, sperm will leave the epididymis and go up this pipe called the vas deferens, or some people call it the ductus deferens. So that's your vas deferens. On billboards all around Houston, you often see where they'll do a vasectomy for you. Well, a vasectomy is the cutting and tying of the ends of your vas deferens to prevent pregnancy, to prevent fertilization. So this is the vas deferens, and notice the vas deferens goes to the back side around the bladder. So it goes to the back side around the bladder. On this slide, they've cut it off, but on the next slide, you'll be able to see it. Now, the picture I'm referring to is page 1033. So on the back side, on the next picture, you'll see this. And right here is a secretory vesicle. So the secretory vesicle. Then you have your prostate. And then you have right about here, and is even identified but not labeled, is the bulbourethral gland. Some people call the bulbourethral gland the Cowper's gland. Okay, so it's sometimes referred to as the Cowper's gland. Now the three that I've mentioned, the secretory vesicle, the prostate, and the Cowper's gland, they all add to make the word semen. So semen is actually really a mixture of Cowper's gland fluid, prostate gland fluid, and the secretory vesicle fluid. So all three. Okay. So the seminal vesicle fluid, the Cowper's gland, the prostate gland, they're all three combined along with sperm. All three combined. Next picture. Here's the other half. Now I said seminal gland, secretory gland, because it is a secretory gland, but it's often referred to as the seminal vesicle. So on this slide, they've labeled it as the seminal gland or the seminal vesicle. Again, you see the prostate, and now on this side of the picture, they have labeled it for you as the bulbourethral gland. Next slide. So according to the last two pictures, make sure you study page 1093. Again, I call it secretory vesicle. Some people call it seminal vesicle. It is a secretory gland. Next. Okay, this one, don't care too much about. This one, I do. Make sure you have a general feel for this picture right here. Okay. Again, I've mentioned perineum. Perineum is everything basically as a triangle between the legs. Next slide. Okay. Now, this slide, this picture doesn't make too much sense, but I want you to make sure you have this notation down. It's best shown in a later picture. Now, the testes are actually formed higher up in the abdominal wall, the abdominal space, and they're actually descending down. We'll see that also. Next slide. Now, this slide is important. Notice what it says. The descent of the testes during the 7th month. So, 7th month of fetal development. 7th month of fetal development is when the testes will descend. So when the kid is born, when the male child is born, he should have his testes already descended. If it doesn't, surgical intervention is necessary. Otherwise, the child may end up sterile. Okay, so it is important. Next slide. Now here's the picture version of what we have been talking about in terms of descending the testes. Notice it is higher up in the abdominal area, the pelvic area, and then it actually descends down into the scrotum. Okay. So that's something to keep in mind. Next slide continues the story. So it slides down, descends down into the scrotal sac. So it descends into the scrotal sac. Now the term that you saw, this gubernaculum, make sure you study that term. So that gubernaculum, make sure you study that term. Okay, next slide. Here's an enlarged view of that. Next slide. Okay, now, before I move forward, if the testes fail to descend, on page 1033, they talk about a condition called cryptorchidism. So the paragraph on page 1033 below the picture. And I've mentioned to you a moment ago that they have a surgical intervention to help with the descending of the testes if that doesn't happen. Okay. So make sure you read up on page 1033, cryptorchidism. Okay, next we have this slide called the spermatic cord. So we're now looking at the accessory structures. Spermatic cord. Now what do I find in there? This slide right there is a good test question. What would I find within the spermatic cord? We find the vas deferens, also called ductus deferens, blood vessels, nerves, and the lymphatics. So this line is very important. Now, you may have heard that some guys, after they get to 40 plus typically, they have what looks like a bag of worms in their testes, okay, in their scrotal sac. That's because the blood vessels are not, they're getting narrowed. And so what they do is they go in there and they open up, they tie up other blood vessels, which improves the blood flow in the scrotal sac. And that improves the quality of the sperm production past 40 years age. So around 40, 45, you see a varicocele repair done on guys. That's just to help them with their sperm production. Now, on page 1035, they talk about inguinal canal, and that's where we're going to be headed to in a second. Okay. So this is the slide that is important. We've already mentioned what is inside of the spermatic cord. It's repeated again here. And it also talks about the inguinal canal. This inguinal canal in guys is very weak, and it makes you very prone to a inguinal hernia. Since that area often gets a lot of pressure and push, so if you have a lot of constipation or you're lifting something heavy inappropriately and you're not bending at the knees like you should, that passageway can tear and lead to an inguinal hernia where your intestines are poking through. That is mentioned on page 1035. Okay. So that is a weak spot in the guys. Okay, next slide. Now we talk about the blood vessels. So we have these blood vessels listed along with the nerves. Now, this pampiniform plexus, that is an important term to keep in mind because it's going to deal with a movement of blood flow very similar to the counter-exchange system that you saw in the urinary. Now, what does it do for you in this case is it moves the hot blood, basically, and cools your testes. So it's actually circulating or air conditioning your scrotal sac. So this is a blood vessel cooling system, very much similar to an AC. And that's actually mentioned on page 1035. Next slide. Okay, we've already talked about the herniation. That's mentioned on this slide. So the inguinal canal herniation is mentioned on this slide. Next slide. Okay, now on this slide, you can actually see the picture version of what goes inside the spermatic cord. So if a surgeon is working within the spermatic cord, the surgeon has to be very careful not to mess with that genitofemoral artery or the genitofemoral nerve. Okay, make sure that they don't mess with any of that. Next slide. So this picture again is an important one to keep in mind for test purposes. Here's the other side of it labeled for you. You now actually see the ligaments. So you see that inguinal ligament, and you actually see the area where they call that weakness in the wall. So that superficial inguinal ring, that is often the weak spot. And that's where the hernias often happen for guys, right at that superficial inguinal ring. Okay. Now, On this slide, they mention the cremaster muscle and the dartus muscle. Make sure you know the functions of both. So make sure you know the functions of both. Now, to some degree, it says the dartus muscle elevates the testes and causes the characteristic wrinkling of the scrotal surface. Let me read that again. So the dartus muscle elevates the testes and causes the characteristic wrinkles of the scrotal surface. Then we have the other muscle called the cremaster muscle. The contraction of the cremaster muscle during sexual arousal or in response to decreased temperature tenses the scrotum and pulls the testes closer to the body. So as you can tell from both the cremaster muscle and the dartus muscle, temperature of the scrotal sac is important. You don't want it too cold and you don't want it too hot. It has to be like Goldilocks, just right. So the temperature of the scrotal sac is maintained partially by the cremaster muscle and the dartus muscle. Next slide. Okay. So the information I just provided you about the testicle temperature is mentioned on page 1035. So page 1035. The author mentions something very, very similar to the Seinfeld episode with Costanza. So if you are a big fan of that show, you might remember Costanza with the cold swimming pool. So let me read you this from the book, page 1035. Sudden cooling of the scrotum as occurs during entry into a cold swimming pool results in contractions that pull the testes closer to the body and keep the testicular temperature from falling. Okay. So if you remember that show, you might remember that. Okay, next slide. Okay. So we have scrotum and position of the testes. Okay. This slide, don't care too much about. Next slide. This one, make sure you have looked at it. Don't care too much about it, but make sure you've looked at it. Next slide. These two muscles we've already mentioned. I mentioned them a little bit earlier, so make sure that you have these two down. So the dartus muscle, the cremaster muscle. Okay. So the dartus muscle, cremaster muscle. Okay. Temperature regulation, you already know why now. So I've given you that information. Now it turns out the key reason for that temperature regulation is the sperm development. So the sperm requires a right level of temperature. If it's cold, then it uses the body's heat and therefore it moves towards the body. If it's already too hot and you need to cool it, it'll move away from the body. So sperm quality and sperm development are highly dependent upon temperature. Next slide. Okay. This slide I want you to write down. It best shows up in a picture form. So you're not going to understand this picture until you look at it later. So this picture and the other picture I had up earlier. So this one and this one is best seen in picture form. Next slide. Okay. This is a very important picture. This is found within the testes, and you have what's called the lobes or lobules. Inside of these lobes or lobules, you will find very tightly coiled seminiferous tubules. So inside of the lobules, you'll find seminiferous tubules. So you have a lobe, and within the lobe, you have a tube system called seminiferous tubule. They produce the sperm. The sperms are actually launched in sort of a reverse way, like a ship would be launched. And I have a picture coming up to show you that. So the way a ship would be launched In reverse is the way the sperm are launched into the lumen of the seminiferous tubule. So the sperm are made and launched in reverse into the lumen of the seminiferous tubule. From there, the seminiferous tubules join at what is called the rete testis. So that's where they're joined. Again, these terms are best seen in picture form. Next slide. This is where all of them are joined. It says connects the rete testis to the epididymis. Again, they will not make sense until you see the picture. So the rete testis is joined to the epididymis by the efferent duct. So now you can see a pathway forming. Next slide. Okay. Here's the picture to keep in mind. Here you have your lobe. Inside of the lobe, you see these seminiferous tubules. So all of this wad of spaghetti-looking material, is your seminiferous tubule. It is labeled for you on the side here. And all of these seminiferous tubules, inside of the tube, you have a thick lining. The thick lining of the tube is where the sperm are made. So you have a tube here. Inside the tube is a lining, and that's where they're made. Now, we said a moment ago that these seminiferous tubules, they all lead to the rete testis, and that is shown here as well. So here's the rete testis, here's one as well. Now the rete testis is going to lead into the epididymis, which would be this piece. But before I get to the epididymis, you see 1, 2, 3. Those are your efferent ducts. So rete testis right here, and the 1, 2, 3 efferent ducts leading to the epididymis. Here's your lining. The terms tunica and tunica always refer to a layering or lining. You see the outer margin and then the inner margin. So they're labeled for you here, the inner margin. Next slide. Here's another view of that. Next slide. Okay, now, this is now looking into the surrounding area around the tubes. So you do have tissue around your tubules. So you do have tissue around the tubules. That tissue space is called interstitial cells. Some people call it the other name, which is the Leydig cells. So outside of your tubule system, you have the interstitial cells, which are the Leydig cells. Now look what they do. They produce the androgens, especially testosterone. So the interstitial cells or the Leydig cells will produce testosterone in the testes, but between the tubes or around the tubes. Next slide. Okay. Now, don't care for this. I'd rather have you see it in a slide. Now, the terminology here is worth noting. Spermatogenesis is the production of sperm, and that makes sense. Okay. So spermatogenesis, the production of sperm. The rest of the slide is best shown in a picture that's coming up. This slide you should keep in mind, because what are we doing in spermatogenesis? What we're really doing is breaking down by way of mitosis, and then what? Meiosis. Now, meiosis or meiosis is going to produce the haploid number of chromosomes in the sperm. So, our body cells have 46 chromosomes. The sperm, on the other hand, is going to have 23 chromosomes. Now the magic of the 23 chromosomes, because of meiosis or meiosis, is that the 23 in each individual sperm will be 23 chromosomes that have been shuffled. Meaning you could have gotten dad's chromosome number 5, whereas mom's chromosome number 10. Okay. So when you get a sperm made, each sperm is going to be genetically different. Okay. So your sperm as the son will not be the same sperm that the dad had. Likewise, each sperm is unique. That's what meiosis does, genetic variation. So you're not your dad and you're not your son. Okay. Next slide. So the story I just gave you is mentioned here. Okay. So this is worthwhile keeping in mind. Next slide. Now, this is where you're going to really study the picture. These are your seminiferous tubules. Okay. Seminiferous tubules. Now, the seminiferous tubules have this thick lining that you see. That thick lining is producing the sperm. In this one, you actually see sperm inside the center. On this one, it says containing late spermatids. So you again see sperm here as well inside. This one is still empty. Now these sperms that are in here were actually launched from the wall into the center. And of course the center being called the lumen. So the walls of these seminiferous make the sperm, and then they're launched in reverse into the lumen. Next slide. Here's an enlarged view so you can see it better. And the author has done a good job with this one. So here we have our lumen of the seminiferous tubule. And we see the wall here. In between these seminiferous tubules, you see the interstitial cells that I've already told you about, also called Leydig cells, producing testosterone. So that would be in between here. So you can see some down here as well. They would be the Leydig cells or the interstitial cells. In between, you see nurse cells. Let me go ahead and talk about that. The nurse cells, their job is to help the sperm while it's being formed. So it's sort of a nurse that helps you grow up, like a nanny basically. So this nurse cell or nanny is going to help you with the development of that sperm. It's going to protect that sperm. And when that sperm is ready to be launched, it'll be launched in reverse. You can barely make out the flagella of some of these. Okay. So they're launched in reverse. Okay. Next picture. Here's the artistic version of it. Done a good job here. The artistic version shows you the interstitial cells, the Leydig cells again. Shows you how spermatogenesis is occurring here. And now you see the sperm with the flagella, and notice all of the flagella are going into the lumen because they are actually launched in reverse. Next slide. Here's the enlarged view of that. Okay, so you see meiosis 1 mentioned, and you see meiosis 2 mentioned. And of course, you should know by now that meiosis is a two-step process. So you have a meiosis 1 and a meiosis 2. And meiosis 2 resulting in 4 haploid cells. So 4 haploid cells, each sperm having 23 chromosomes. And each chromosome or rather, maybe back up, each sperm will have 23 different chromosomes. So there's a shuffling in the male and a shuffling in the female as well. Okay, draw a line. Okay. Now before I move to the next slide, do you see this dotted line right here? That dotted line is called the blood-testes barrier. It's the blood-testes barrier. As you can see, after this dotted line, there are no blood vessels here. All the blood vessels are shown here. The reason for the blood-testes barrier is blood is dirty. And whatever germs you might have in the blood, very similar to Biology 2401 with the blood-brain barrier, we have a blood-testes barrier, preventing germs from infecting this region, infecting the production of the sperm. So you never want to break that blood-testes barrier, otherwise you're going to have a problem with sperm development. Radiation, some chemicals will actually pass the blood-testes barrier and affect the sperm development. Okay, so you have to be careful about the blood-testes barrier. Next slide. Now earlier I gave you this term, nurse cells. They're now called Sertoli cells. So as of this slide, nurse cells are also called Sertoli cells. So nurse cells, Sertoli cells. And what they do is pretty much nurse and guide the sperm, pretending as if the sperm are newborn babies and the nurse is guiding them like a nanny, helping in their development. Okay, next slide. We've already talked about this, so we have a little bit of mitosis initially, and then meiosis, and then we have spermatogenesis. Next slide. Now, mitosis, the initial part is making two diploid cells, but I don't want you to get confused by that. I really want you to study the fact that they actually become through meiosis or meiosis. This is just to let you know the difference. So if you don't know the difference between mitosis and meiosis, yes, you should review this. So this slide is really for the review of someone who does not know the difference between mitosis and meiosis. Okay. This slide again will refresh your memory if you don't know the difference. Now I already told you that each sperm, when they're finally done, will have 23 chromosomes, half the number that you normally have in the rest of your body with 46 chromosomes. Next slide. Again, don't care for that one. We've already talked about this. I gave you this answer earlier. Meiosis will produce 4 haploid cells, each with 23 chromosomes. The slide fails to mention the fact that the 4 haploid cells or the 4 haploid sperms are genetically different. Next slide. This is again a rehash of general biology. So if you didn't learn this, it is important to keep that in mind. Now the reason why this is being brought up as a refresher is to let you know that because in meiosis we have crossing over, we're going to get a genetic variation. Remember I said, you are not your dad. You are not your son. Okay? So you have given certain traits to your son, just as certain traits were handed to you by your father. So father, son, grandson. But there's going to be a genetic variation. So they're never the same. Continuation, continuation, continuation. Now, the author here is letting you know that it's a reduction division because meiosis starts off with 46 chromosomes and ends up with 23. So that's why it's called a reduction division. Next slide. So this is now another refresher on the differences between meiosis and mitosis. Here's the point that I was making. Your sperm will now have 1, 2, 3, 4. This is one sperm, another, and another, another. So there's 4 sperms basically shown here. And each sperm will be genetically different. Okay. Okay, moving to the next major slide. We've already talked about this as well. Next slide. We've already talked about this as well. Okay, now, this slide is important. So this slide is important. 6 major functions of the Sertoli cells or nursed cells. Earlier I told you that it protects or guides or acts like a nanny to the new developing sperm. That's because it maintains the blood-testes barrier. That's why it's like a nanny, a protection to the sperm. It supports spermatogenesis. It secretes MIF. It is a very important thing that you know what MIF is. So definitely look up the Mullerian inhibiting factor, MIF. That is very, very important. Next slide. Okay. We've already talked about the blood-testes barrier, so we'll move on from there. Now, here's the other thing that goes on. Nurse cells are stimulated by FSH, testosterone. Now, are you noticing a storyline here from chapter 18? So FSH, testosterone, will stimulate nurse cells. Next slide. So this slide is important. Next slide. This is what the nurse cell will do. And we've already said it protects that sperm, provides the right nutrient and chemical environment. Okay. Next slide. Here are the hormones that were mentioned on the previous slide. Now, this one is a very important one. Here's why. Look what it says at the bottom. Inhibin depresses production of FSH. So depresses FSH. It depresses secretion of GnRH. So we actually have a feedback system with this. Many books will have a picture coming up shortly on this. Next slide. So this slide is important. The action of inhibin. Okay, this was the other one that was mentioned. Okay. Notice it says androgen binding protein binds androgen, especially testosterone. Stimulates spermatogenesis. Production of APB, or androgen binding protein, is stimulated by FSH. So you should be starting to make some kind of a flow chart, and that's what we'll be looking at shortly. Next slide. Now this is the one I said that you have to make a note of and look at. Here's why. Secretion of Mullerian inhibiting factor. Secreted by nurse cells in the developing testes. That was mentioned already. Look what it says. In males, inadequate MIF production leads to retention of ducts and failure of the testes to descend. Okay. Look what it says up here. Helps form uterine tube and uterus in females. Okay. All right. So you see that there is a difference. It is this MIF that makes a difference between females and males. So if this is not working properly, you're going to see a problem with the testes being formed and the testes descending. So you're really built to be a female as a basic model. And then it is the difference in the Y chromosome providing this information. So the Y chromosome provides the hormone information to make testosterone, MIF, and all of that. So let me say that one more time. It's the Y chromosome that's going to play a role in production of this Mullerian inhibiting factor, which plays a role in the development of the testes and the descending of the testes. Okay, next slide. Okay, now the author on page 1041. So the author on page 1041 is going to go into what a sperm looks like. So what a sperm looks like on page 1041. Okay. So make sure you have these pieces in mind, and we're now going to look at each one of them. Next slide. This slide is important. So we have the head containing the acrosome. So the head containing the acrosome. So the sperm has a head containing the acrosome. Of course, you'll find the nucleus there as well. So the nucleus is inside, within that acrosome, within that head. Okay. So the nucleus is there, containing 23 chromosomes. The acrosome contains enzymes necessary for fertilization, one of which you already learned in Biology 2401, and that was hyaluronidase. So among enzymes, one of the ones you've learned is hyaluronidase. Okay, next. Midpiece. This contains your mitochondria. Why would you want to have a lot of mitochondria there? Because you have to have it to move that flagella, that tail that you see on page 1041's picture. Now, of course, by mitochondria, we need some type of energy source, and that energy source is going to be fructose, and we'll see that later. So the energy source will be fructose to help produce ATP by way of this mitochondria. Next slide. Here's the tail that we've mentioned. So the tail, the flagellum. And here's a picture of one on the next slide, page 1041. Now, one of the things I want you to understand from this picture, I'll go ahead and go to the next one so you can see the final version. One of the things you need to understand about this is that the sperm has about a 48-hour to 72-hour type of lifespan. It doesn't live long. Typically, the answer would be around 2 days. So the typical answer is about 2 days. It can extend to 3. So this thing only has a 2 to 3 day window to get to wherever it needs to go. Okay. Again, the energy source will be fructose to get up there. Okay. And the hyaluronidase and the enzymes that are necessary for fertilization will be in this acrosome, this tip. Now the way I look at it is like how many licks does it take to get to the center of a Tootsie Roll lollipop? It's exactly that same process. As the sperm loses the surface of that coating, it releases the enzyme. Now, will one sperm be enough to break into the outer covering of the egg in the female? The answer is no. Okay. It will take several 100,000. So several 100,000 sperm to have enough hyaluronidase and other enzymes to break into the egg's outer covering. And that's not going to happen with just one sperm. And that is why the amount of sperm, or the number of sperm that have to reach the site of fertilization, has to be in the 100,000 range. That means if a guy already has very few sperm and they're not enough to survive through the entire vaginal and up into the uterus, and it's not going to happen, well, that's why he ends up being sterile. So he may have some sperm, but not enough to make it through the journey, and not enough to get to the egg and still have enough sperm to produce the ability to penetrate and fertilize the egg. So that story is being brought here to compare what happens now to later. Okay. Now, this slide right here, I've already mentioned the bottom portion. So I've already mentioned the fructose story. This general idea up here is worthwhile knowing. Not too important, but it is worthwhile knowing. Next slide. Sperm maturation. Look what it says here. Testes produce physically mature sperm that cannot fertilize an oocyte. Okay. So sperm maturation. So once you have the testes producing the sperm, they're not ready to fertilize yet. So where will they go? They'll go to the epididymis. And in the epididymis, they will become functional. They'll receive their nourishment, storage, and transport. And that is why they go off to the epididymis. Next slide. I've already mentioned it. Here it is. Are functionally immature at first. So what happens? They go to the epididymis to become capable of locomotion, capable of fertilization. And of course, how do I get there? By way of the efferent duct I showed you before. So from the rete testis through the efferent duct into the epididymis. Now are you noticing that that's coming up over and over? So the pathway is important. So you're going to find the pathway to read something like this. The seminiferous tubules, straight tubules, rete testis, efferent ducts, and then the epididymis. Next slide. We're now at the epididymis. Okay. Don't care for too much on this one. This one is important. So this is where the sperm will be stored and receives its ability to do things. Now in the epididymis, that's where they're going to be held before ejaculation. So that's where they're held before ejaculation. Now, this is where you can screw up. So if you have some bad sperm, you want to get rid of that. So if you have a flu, a cold, and you've made sperm during that flu season you had, when you had the cold, when you had the flu, that sperm is not in the best capacity. So where will it go? Once you've made them, they'll go in the epididymis. So that lot is a bad lot. You don't want to use that lot because you don't know how bad it is. It could be the defective sperm, defective chromosome, whatever it is. So you wait, you expel that and you wait another month or two, and then you have a new stock in the epididymis, and that stock will be a healthier stock. Okay. So whatever is in the epididymis is what you'll be using. Next slide. Okay, here's a picture version of what I've said so far. So here you have the seminiferous tubule shown here as well. Then you have this line right here. That piece right there is the straight tubule, rete testis, and then you have the efferent duct going into the epididymis. So we have the body portion and the tail portion, and then it leads into your vas deferens. And this whole ring right here, as we saw earlier, would be the spermatic cord. Okay, next slide. So this is where they become functionally mature. So we've already talked about this. This one is worthwhile writing down. Monitors and adjusts the fluid produced by the seminiferous tubule, recycles damaged spermatozoa, and stores and protects spermatozoa. Now you see why I said that if we have a lot of damaged ones that have not been recycled, that's what's going to come out of you. Now true, some of them will be reabsorbed into the body tissue and will be disposed of. But again, sometimes it doesn't work so well. So the object of the game is just get rid of it and make a new stock. When I go to the grocery store and I see some can of beans or whatever, and I read the label, I always go to the very back because that's where the fresh stock is. The newer dates, the better dates are towards the back because they always replenish from the back. Same concept here. Next slide. Now, look what it says here. This one's important. Are mature but remain immobile. To become motile and functional, spermatozoa undergo capacitation. That is the stripping off at the top. So stripping off at the top. Let me read you this line from the book. To become motile and functional, spermatozoa must undergo a process called capacitation. Capacitation normally occurs in 2 steps. Spermatozoa become motile when they are mixed with secretions of the seminiferous gland, and 2, they become capable of successful fertilization when exposed to conditions in the female reproductive tract. Now this line came from page 1042 underneath the picture. So page 1042 underneath the picture explaining what capacitation is. Notice it says they become capable of successful fertilization when exposed to the conditions in the female tract. Okay. So it's going to happen before and after it's into the female tract. Next slide. So this slide right here came from page 1042. Next slide. The vas deferens, or the ductus deferens. Okay. Don't care for that slide. Okay. This slide I do care for. This is important. Ductus deferens is lined by ciliated epithelium. So you get a peristaltic contraction. Very similar to the ureter story before. So you have a peristaltic contraction process. Can store spermatozoa for several months. Okay. Next slide. Now we're continuing our story. I've already mentioned that the vas deferens goes behind the bladder. So we're looking at the back view here. Goes behind the bladder and it enters the seminal vesicle or the seminal gland on the back side. Then you see where it says ejaculatory duct. So the seminal vesicle, seminal gland will release its secretions along with the sperm. So the sperm will come down the vas deferens. The fluids of the seminal gland or the seminal fluid will come out and it will go through the ejaculatory duct right into the prostatic urethra. And yes, the prostate will also do what? Add its fluid. And then when it gets down to right about here, where you would find the membranous urethra, you will have the bulbourethral gland or Cowper's gland mentioned before, adding its components. Next slide. Don't care for that one. We've already mentioned it. Don't care for that one. We've already mentioned these three. prostatic, membranous, and spongy. So prostatic, membranous, and spongy. And they are in the correct order. So prostatic, membranous, and spongy. Next slide. These three, again, this is the second time we see that. We've seen these three at the beginning of the chapter. Notice we're now using the word semen. So semen is a collection of sperm and the fluids from all three of these that you see in front of you. So semen is sperm plus fluid of the seminal gland, prostate gland, and the bulbourethral gland. Next slide. Don't care too much for this picture. Next slide. Now, I do care for this. Look what it says here. Produces about 60% of the semen volume. That is important. So the seminal gland will produce 60% of semen's volume. Next slide. We have the seminal fluid now. This slide is very important. I'm going to go through it line by line. First, it says high concentrations of fructose. I've mentioned fructose before. Well, this is where it is coming from. It's coming from the seminal vesicle fluid. Okay. Next, prostaglandins stimulate smooth muscle contractions in the male and female. This is to allow two things. One, to allow the fluid to leave the male, but at the same time cause the female to pretty much have contractions that suck up the semen once it is in the vaginal tract. So it actually causes the female to squeeze and causes the fluids to move deeper into the female tract. Next, fibrinogen forms a temporary clot in the vagina. And that's going to cause a thickening of the semen once it's deposited into the vaginal tract. So it's going to cause a little thickening. Next we see is slightly alkaline. Okay. Now why do I want that? It says it nicely here. To neutralize the acids of the prostate gland and the vaginal tract. The vaginal tract is very harmful for sperm. It's very acidic. It will kill it. So the first thing we got to do is neutralize the pH of the vaginal opening. And that's what this fluid will do. Next slide. Okay, it continues. Now this one I'm going to let you go look at, but notice it says contractions are controlled by sympathetic nervous system. So yes, things down there are really controlled by things up there, if you know what I mean. Now, having said that, next slide. Okay, repeated the picture again, and we're now moving to the next picture, the prostate gland. Now the prostate gland, there's not much mention on this slide, so we're going to go to the next one. And this one is important. Okay. Prostatic fluid is slightly acidic. It makes up about 20 to 30% of the semen volume. Contains an antibiotic called seminal plasmin. Is ejected into the prostatic urethra. And again, we have peristalsis involved in squeezing and moving the fluid forward. Okay, next slide. Again, we see the same picture again. And now we have the Cowper's gland. We already know where the Cowper's gland is, so I'm going to move to the next slide and tell you what it does. The Cowper's gland, or the bulbourethral gland, secretes a thick alkaline mucus. This is a thick fluid. Helps neutralize the urinary acids in the urethra. That is both in the male and the female. So if there's any urinary acids in the urethra, this will neutralize it. During intercourse, it actually lubricates the tip. So it helps lubricate the tip during intercourse. Next slide. Again, we've already seen this picture. Next slide. Now, we already know what semen is. How much? About 2 to 5 mils. Now, here's what it says: abnormal low volume may indicate problems, either with the prostate or the seminal gland. So if you've got low production of semen overall, that might be a prostate gland problem or a seminal vesicle problem. The other issue is if you have less volume, chances are you also have what? Less of a count. Next line down here is taken of semen collected after 36 hours of sexual abstinence. Okay, so that's when the count's taken. Now, look what it says here. Normal range is about 20 to 100 million per mil. Now, if we take this top number up here and find an average, say about 4. 4 would be 4 mils times 100. That would be 400 million per semen ejection. 400 million. If we take an average of about, say, 60, 60 times 4 would be about 240 million. Okay, so to give you an idea. Next slide. So remember, how much do we have to reach the fertilization point in the female? Several 100,000. So that must be for a reason why we have so many millions worth. Because a lot of them are defective, a lot of them get stuck in the wrong places, and they never make it to the fertilization site. So that's why we have so many. Next slide. Now, these are the things that we've already talked about summarized. Now, one of the things I want you to get out of this picture, especially the bottom, especially this part, it turns out that all of these ingredients at the bottom are designed to package the semen in a thick mucus. And it has a time period by which that thick mucus needs to loosen up inside the female tract. Now that time is called liquefaction time. So if for some reason the liquefaction time is longer than half an hour to an hour, then what will happen to that sperm? They will die because they are not moving anywhere. They're stuck in this thick mucus that has been deposited into the female. So liquefaction time is one of the factors that they check for. The other thing that they check for is once the liquefaction time has passed and the sperm are now free, they check how fast the sperm actually move, almost like a race, and that is called motility. So they check for parameters like that. Next slide. We've already talked about this, so I don't care for it. Next slide. This is important. So I want to make sure you have this slide written down. So make sure you know the 3 parts listed here. Next slide. So this one's important. Next slide. On this one, make sure you are able to identify these. So this slide is something that you need to be able to do on a lab day. Now the author has labeled membranous urethra for you here. So make sure you're able to label this one on a lab day. Next slide. We have the penis. This is another one that you need to be able to label on a lab day. So you have the 2 main chambers shown here, which are the corpora cavernosa. Then you have the one down here called corpus spongiosum. So make sure you know that. So corpus spongiosum and the corpora cavernosa. Next. Now let me back up for a second. Notice you see blood vessels and nerves here. These play an important role in the blood flow through the penis. And drugs like Viagra and all of that are designed to do what? Increase the blood flow into the penis at the time of intercourse, therefore a erection. Next slide. Here's another one that you need to be able to appreciate. So it gives you a pelvis orientation. Okay, so this one's important. Next slide. Okay, don't care too much about that one. This one I do care for. This is the foreskin. Some countries, some cultures, they have circumcision. So circumcision can help prevent infection because it actually removes the covering that goes over the head of the penis, and therefore it is cleaned out. It prevents infection. Some people even say it prevents the penile cancer. So that's something to keep in mind. So this slide is important. Next slide. This one, don't care too much about. I've already mentioned the blood vessel issue here, so that's already been mentioned. I've already asked you to know these. I don't care for that. I've already asked you to know that one. Now comes hormones. These hormones you had in chapter 18, they are still important. So you have the FSH in its story, LH its story, and then of course GnRH. Okay, so those are important still. Here's how they play a role. GnRH plays a role in controlling the rates of FSH, LH, and then testosterone released in response to LH. So LH goes with testosterone. You of course already know where it comes from. So FSH, LH. Next slide. Okay, this was mentioned earlier, but is again restated. FSH and testosterone, we know that it targets the nurse cells, and the nurse cells in turn promote spermatogenesis, and they also secrete the androgen binding protein. So this is a slide previously mentioned. Next slide. We have a feedback system here. So that's what we're getting into. I did mention that books often talk about the feedback system. It's coming up. So this is a feedback system to keep in mind. Spermatogenesis is regulated by these three. As spermatogenesis accelerates, inhibin secretion increases. So this is a way that you're going to regulate how much of your sperm you're producing. So if you've got far more sperm production than you need, inhibin is going to rise. And inhibin will have a negative feedback on the other two. Let's look at the slides and then we'll see a picture related to that. So inhibin inhibits FSH production. It suppresses GnRH. So if you've got far more sperm production than you need, then inhibin is going to tell FSH and GnRH to stop it. It's almost like if you have far more cars made by the manufacturer, the dealer is going to say basically what? I don't want any more cars from you. I need for you to stop making them so I can start selling the ones I've got. It's very similar to that, a negative feedback. Okay. Look what it says here. Elevated FSH will increase inhibin. FSH will decline, and then inhibin will stop. Again, this is a negative feedback system. Okay. Luteinizing hormone will go with the Leydig cells. So LH will go with the Leydig cells in terms of testosterone. Next slide. Now, this is a slide that you must keep in mind because it falls out of the place of many other chapters. It doesn't have a place to be other than here. This is simply letting you know what testosterone does for you. One of the things I want to keep in mind is from the bottom up here, muscle growth. That's why they take steroids for muscle growth. Then comes blood cell formation. So it actually improves blood cell formation, improves muscle growth, and improves muscle protein synthesis. And that's why testosterone, the rise in testosterone is related to rise in your muscles as well. So if you exercise and you boost your testosterone level, you're going to boost your muscle growth, your blood cell formation, and your protein synthesis for the muscle growth. Of course, the other advantage is testosterone will rise in your libido, so you'll have a sexual drive. So these are all things that relate to testosterone. Testosterone also deals with your secondary sex characteristics, such as facial hair, increased body mass and body size, characteristic of adipose tissue deposits. That is why unless a female takes bodybuilding steroids with testosterone, a normal female working out with weights and all cannot become like a big, heavy Arnold Schwarzenegger type of person. It won't happen because they do not have the degree of testosterone they would need for that to happen. Now, if you're a bodybuilder who is a female taking steroids like this, then it'll happen. Next slide. Now, this slide here is page 1047. Again, the author has broken it down into two parts, the bottom and the top. Much of what I just went over as slides is now summed up on these two pictures. So definitely study page 1047. Okay. Now, this is something you've already learned before, so I'm going to pass that up. How testosterones work. Remember, they are steroid hormones, and therefore they're able to go straight into the DNA and have an effect since they're lipid-soluble hormones. This story here is now going back to explain something I mentioned before. Where will this testosterone come from early on in the production? It comes from the Y chromosome. So the Y chromosome is what's going to make this slide happen. I would make a note of that. So it's the Y chromosome that's making this slide possible. If you want a slide number, it is slide 135. And this is the one that's because of the Y chromosome. Next slide. These are all because of what? The Y chromosome. Next slide. Okay. Now, we do have females and males have a form of estrogen. So this is a small version of that. Next slide. We're now going to continue with the female reproductive system for chapter 28. Now, first of all, I really don't care too much about this slide, because we should already know by now that females produce sex hormones as well. They have gametes as well, but in the terms of gametes, we call them the eggs or the ova, and their job is to protect and support the developing embryo within the uterus. So this is a general slide. Next, we have our parts. And as I said before, all of these parts you should be able to identify on lab day. Next slide. Here we have the female version, sort of like the male, but the female version of page 1050. Again, you should be able to relate this to the lab model. We actually have a model like this, so be able to label it on the model. Here's the the anterior part, and here's the posterior part of it. Now in this picture, you outright can see that the uterus is sitting on top of the bladder. So here's your vaginal area. So that's the vaginal area. This is now the bladder and then the uterus. So when the baby is pushing down, it's pushing down on the bladder. Okay. Now, you also see words on the other side that relate to things like uterus. Now, in the male portion, I mentioned something in passing, and that was the uterus has a thick muscular lining called the myometrium. So we have a thick muscle lining called the myometrium. That's what's going to squeeze at the time of uterine contractions. Likewise, we have the endometrium, and that is what the fetus is going to live off of. So when the embryo comes in, burrows its way in, it burrows its way in into the endometrium. And it's within that endometrial lining that is going to form the placenta. So we'll see that. So again, make sure you're clear on this picture and that picture for lab purposes. Next slide. On this slide, I want you to understand that it's the ligaments that actually hold the female parts in place. So it's holding the female parts in place. Next slide. Here we have an important picture, page 1051. In this picture, we see the vaginal area at the bottom, the vaginal folds called the rugae, the cervix, the opening of the cervix, so the cervix and the opening of the cervix, the main portion of the uterus. Again, you see ligaments holding all this in place. You see this big area here, also a ligament called the broad ligament. Here's an ovarian ligament holding the ovary in place. And then you see the fallopian tube or the ureter tube. The ureter tube ending with finger-like projections called the fimbriae. Again, you see suspensory ligaments holding all this in place. Now, these fimbriae are not directly attached to the ovary, but they are actually creating a current. And that current is going to guide the egg that is released from the ovary and is going to guide the egg into the fallopian tube. So if that egg ends up out here somewhere, it's going to disintegrate and then eaten up and chewed up. So it won't do anything. But the egg's purpose is to be guided by the current of the fimbriae into the fallopian tube. And right roughly where this is labeled, roughly where it's labeled, is called the isthmus of the fallopian tube. So roughly right here, the isthmus of the fallopian tube is where you're going to see fertilization typically. So the isthmus of the fallopian tube. So it's a long journey to get all the way up here. And remember, the sperm have about 2, maybe 3 days tops. Next slide. Here's the opposite side. Pretty much the same story. Next slide. Now, this picture right here on page 1051 is a very important picture. Now, I'm not caring too much about that side of the story, although it is important, but this side of the story is very important. Now, in the female, as I mentioned at the very start, the female is going to have most of the story when it comes to reproduction. I would say something on the order of 80 to 20. 80% female, 20% male. Now, in this 80-20 story about female being the more important one or the more valuable one overall, it's because it has 3 levels of stories here. The first level is what goes on in the ovary itself. So you have one story of what goes on in the ovary. Then you're going to have another story that goes on with the hormones. And then the third story is what's going to go on the endometrial lining of the uterus itself. Now, Nemish, is there a chart later in the chapter relating all of these 3 stories? You bet there is. The problem is you need to understand that all 3 stories are interrelated. So what goes on in the ovary is because of hormones. Whatever happens in the ovary is going to play a role with the endometrial lining. Now, I'll also be mentioning days. These are typical day numbers. We're assuming that the female has a typical 28-day cycle, and that is the overall cycle. So 28-day cycle, the overall cycle. Now, here we see these little primordial follicles, and you'll see a better picture later, these primordial follicles, and they're actually being developed. You can see them here too. So here we are having a primordial follicle develop into this mature follicle. Now, what happened here, not shown in this picture, is that as this primordial follicle, shown there and here too, as these primordial follicles developed and it turned into a mature follicle, it actually sped out a secondary oocyte. Not shown here, but it's mentioned. A secondary oocyte was released. Then, as we go around the cycle, we get this thing here, corpus luteum. The corpus luteum is what we have as a leftover of the mature follicle. So the mature follicle releases the secondary oocyte, which we later will call ova, and it turns into corpus luteum. The corpus luteum will play an important role in maintaining the endometrial lining by way of progesterone and estrogen. So Nemish, are you telling me that this developing follicle is actually producing progesterone and estrogen? Yes. So it's the ovary that's producing the estrogen and progesterone. Now, who's causing the follicle to develop? FSH, follicle stimulating hormone. Now, Nemish, are we going back to chapter 18 with FSH, LH, GnRH? You bet. So just like in the male, we'll see it again. Next slide. Okay. So all that I've just said is now found on this slide. You can actually see it. Production of oocyte. That's what the ovaries will do. Secretion of sex hormones, estrogen, progesterone. So I've mentioned it in the picture a moment ago. It is now written for you. Secretion of inhibin involved in the feedback of FSH. So we see that feedback system again, and you'll see a similar picture as you did with the male. Next slide. These are terms I want you to be able to relate to, but I don't care too much about them. Especially the one I do care for is the ovarian ligament, because I've already talked about it. So ovarian ligament is important. Okay, this slide, don't care too much about. This slide, don't care too much about. Okay, this slide is important. It's an overview. Now, this one is important because, unlike in the male, there's actually a little freeze or a changeover. Notice it says, begins before birth, but then accelerates at puberty. So it turns out that the eggs, this primordial egg that I spoke of, it is actually frozen in the middle of meiosis. So some of that meiosis happens when the baby, the female, is born. And then the rest of the meiosis happens at puberty. Okay? So you see that there's a starting point and an ending point, and this time a freeze in between. Here's a picture version of what I'm getting at. We're only going to talk about this guy here. Don't care about that one right now. So here's what we got. Okay. So here's our meiosis, and it's frozen, and it says it right here. Meiosis 1 is then completed, yielding a secondary oocyte. But look what it says here. They proceed as far as prophase of meiosis 1, but then the process comes to a halt. Okay. That's what I'm getting at. Okay. So we get up to meiosis 1, prophase, but then the process comes to a halt. Here's the important sentence that you need to continue. So this slide is important. The primary oocyte remain in a state of suspended development until the individual reaches puberty, when rising levels of FSH trigger the start of the ovarian cycle. So whatever pre-primordial eggs you have, they're frozen right at meiosis 1, and then they continue once they reach puberty. So when a person reaches puberty, that's when they continue. This slide is important. Okay. It says, each month after the ovarian cycle begins, at puberty. Okay. So each month after the ovarian cycle begins, one or more secondary oocytes leave. Now, this right here, called the mature ovum, that is your secondary oocyte that has been released from the ovary. Notice it is a haploid. So just like sperm, there are 23 chromosomes haploid. Now, you're going to end up with 1, 2, 3 polar bodies, and they disintegrate, and they don't play a role later on. So unlike male, in the male you had 4 sperm that developed from meiosis. That is not true in the case of the female. In the female, only 1 of them turns into a mature egg. The other 3 disintegrate. So yes, this picture and the one before, these two are important. So your mature egg, your ovum containing 23 chromosomes, will be released from the ovary into the fallopian tube. Okay, so that's your mature ovum. Again, as with the male situation, just as each sperm were different genetically, although they all had 23 chromosomes, it was a genetic shuffle, a genetic variation. That is true here as well. Next slide. Make sure you take notes on the following. Okay, so make sure you take notes on the following. So this slide is important to keep in mind. I've already mentioned the top part. It says, stops at prophase of meiosis 1. When does it continue? At puberty. So it continues at puberty. By puberty, the female has about 400,000 eggs. Now these are not the mature ova. Okay, so they're not the mature ova. They're just the developing oocyte. Okay, so they're just the developing oocyte. So by puberty, the number drops to about 400,000. So this slide is important. Next slide. Okay. At puberty, it's the rising FSH that triggers the ovarian cycle. Now, Nemish, FSH is going to cause every month. Matter of fact, it says it right there, each month. So FSH is going to stimulate the follicle, hence the name, follicle stimulating hormone, FSH. And every time the rise in FSH in the ovary, a new egg will start the process that you saw a moment ago on page 1051. So that process that you saw on page 1051 that I described, going from what the picture shows, egg nest, germinal epithelium, okay, to mature follicle, that bottom train, that bottom portion is happening because of FSH. Next slide. Okay. Go ahead and make a note of this. Okay, so make a note of this. Now look what it says. Meiosis is completed upon fertilization. So meiosis will complete itself upon fertilization. What is released from the ovary? Secondary oocyte. So what's released from the ovary? Secondary oocyte. What happens to the other polar bodies? They disintegrate. Next slide. Now, when we say ovarian cycle, we're talking about what happens to the ovary. So what's happening in the ovary? It has two main phases, the follicular phase and the luteal phase. They're basically saying what happens before ovulation and what happens after ovulation. That's basically how it's broken down. So what happens before ovulation and what happens after ovulation. Next slide. Okay. Don't care too much about this picture. Okay. Don't care too much about this. Now, I call it primordial each time, but you can call it primary oocyte as well. Okay. So primordial follicle, primary oocyte, and I'll be using that term in a moment. I'm now on page 1053. Okay. So we're now looking at a blow-up of page 1053. Instead of going through 1, 2, 3, 4 all the way down, I'm going to use page 1053, the center picture. So I'm going to use this one to describe it. So now you see the primordial follicles developing into primary follicle, secondary follicle, tertiary, and then finally mature, and then it's going to pop. Now this process right here at the top, going from say 9 o'clock to 1 o'clock position, this process is made possible because of FSH. So let's make sure we are clear about that. So this top process right here is made possible because of FSH. Now, as this follicle is developing and growing, so as the follicle is developing and growing, the follicle itself, and this is important, the follicle itself is producing estrogen. So yes, FSH is causing the development of the follicle from primordial to primary, secondary, tertiary, and so on. That's because of FSH. But the follicle itself is producing estrogen. This estrogen will act on the endometrial lining. Okay, that's fact number one. Then you see here the egg has been released. So this is your secondary oocyte released. It will be captured by the currents formed by the fimbriae mentioned before. Now, when this thing matures and turns into corpus luteum, the corpus luteum will produce progesterone and estrogen to maintain the endometrial lining. Okay. Now, a moment ago, I told you that we had two phases. And we used the language pre-ovulatory and post-ovulatory. So you know that this is the follicular phase, and this must be the luteal phase by its very name. So we have the follicular phase and the luteal phase down here. So what do you think the corpus luteum is saying to itself? It's saying, I'm going to produce estrogen and I'm going to produce progesterone until either the egg is implanted or not. And I'll give it about 2 weeks. So now, how am I counting my time here? Roughly around day 14 is when this egg was released out of the typical 28-day cycle. So day 14 is our ovulation time. So day 14, ovulation. So I now have the other 14 days or 2 weeks for the corpus luteum to maintain itself. When those 2 weeks expire and nothing has become of this non-fertilized egg because it never reached fertilization, so the sperm never got there, Friday night, Saturday night never happened, then what happens? The corpus luteum disintegrates. So it only has a typical 2-week span. And the corpus luteum dries out and turns into corpus albicans. So that's around day 28. As a result of corpus albicans, the progesterone and the estrogen no longer maintain the endometrial lining. So from day 28 onwards, which restarts the cycle, so instead of saying day 29, we restart by saying day 1, day 2, day 3, day 4, day 5. And that will be the days of menstruation or menses. So once I have corpus albicans and the estrogen and progesterone go bye-bye, we're going to turn into having menses or menstruation from day 1 to day 5. Now, Nemish, are you telling me that from day 1 to day 5, while menses is occurring in the endometrial lining of the uterus, this whole story is going to reset itself? So day 1, day 2, day 3, day 4, day 5, all of this is restarting again? That's exactly what I'm saying. So by day 1, FSH is having its influence all over again on the ovary. Remember, it is a cycle. Next slide. Okay, we already know about the fallopian tube. Next slide. This slide right here, I want you to keep in mind. I've already mentioned this earlier. The isthmus of the fallopian tube is typically where fertilization will occur. Here's a picture version of this slide. So we see the isthmus of the fallopian tube right about here. And if you recall in the previous pictures I showed, that's where the word ureter tube was labeled. So that's about where the fertilization will take place. So that's where the sperm has to get to. Next slide. So this slide is important for labeling purposes. Next slide. Okay. This is somewhat of an interesting one. By now you should know that it's always lined with epithelial, because anytime you see the word tube, you should know it's epithelial tissue. That should be a Biology 2401 story. Don't care for that slide. Really don't care for that one. And now we move to this one. Now, of all of this, and by now you should know there's ciliary and peristaltic movement, but look what it says here. A few hours before ovulation, we have a turn-on beating pattern. Initiate peristalsis. Normally takes about 3 to 4 days. Okay. So to go from where we had our fimbriae all the way to the ureter cavity, it takes about 3 to 4 days. Okay, so 3 to 4 days from the time we pop out of the ovary all the way to the ureter cavity is about 3 to 4 days. That's going to be an important number count later. Next slide. Do you see numbers here? You see time periods here. Okay. This slide I want you to make a note of. But the most important part of this, in which I've already told you, fertilization occurs where? In the isthmus of the fallopian tube, that's mentioned. But this number right here, it says, secondary oocyte must meet a sperm during the first 12 to 24 hours. That means the egg has a 12 to 24 hour time period. Okay. After the 24 hours, it's not a good idea to get that egg fertilized because it's not a healthy egg anymore. So when the fertilization occurs in the 12 to 24 hours, that's a good thing. Okay? So we're looking at a 2-day time period typically for the egg, and we're looking at about a 3-day time period for the sperm, and we'll see that later. Okay. This slide I want you to keep in mind. So uterus, mechanical protection, support, and waste removal. So just keep that in mind. Next slide. Really don't care for that. Typically, the uterus faces forward, and that's what this is, forward. Once in a while, some female have it flexing the other way, where it bends backwards. Okay, it can happen. Okay. And you use words like prolapse if you ever hear about that, where it prolapses the other way. Next slide. This I've already asked you to know, so make sure you keep a note of that. All the different ligaments that hold that uterus and the ovary in place. Okay. To me, ligaments here are like tent strings, holding the tent in place. Next slide. Okay. Don't care too much about that. The only reason I care for the word fundus is it's the top of the uterus. So it's the top of the uterus. Okay. Next question. Next slide. Cervix we've talked about. We saw it in the picture. And we talked about the external os, which is the opening. Okay. So we've seen that already. Next slide. We've seen this as well. Next slide. Don't care too much about that. Now this part right here is going to come into play later, but I don't care about that right now. Next slide. This is just an FYI to let you know we're looking at what kind of interaction here, parasympathetic and sympathetic. Okay. Now why is that a true understanding here? Because that's where your blocks are given. So when you give an epidural, the epidural is given here, T10 to L1. So that's where the epidural is given. So this is really related to that. Next slide. Okay. This is a picture that we've already talked about and looked at. You now see the isthmus of the fallopian tube. You see a nice layer of the uterus. The outer is the perimetrium, myometrium, and then the inner lining is the endometrium. This is where the fertilized egg will implant. Okay, that's where it's going to implant. Next slide. Here's the other side of it. Next slide. Remember, I've already talked about the ligaments, so keep those in mind. Okay, here's the top view showing the back of the person, the front of the person, and this is how it's usually oriented towards the front. So the uterus is folded forward towards the front. Okay. We've already mentioned this. So myometrium is a muscle layer. Endometrium is glandular because it produces a lot of mucus. That's why when you have the sloughing off during menses, you're sloughing off a lining of the mucosa. Next slide. We've already talked about this. Remember, it has a lot of muscle layers, so you expect that here. Okay. Provides force to move the fetus out of the uterus, so you expect that during uterus contractions. Next slide. We've already talked about that as well. Now, I've already mentioned this, but let me go ahead and show it to you slide-wise. Estrogen causes glands, blood vessels, and epithelium to change on a monthly cycle. So that endometrial lining is actually going to change. Next slide. Here's what's an example of one. All of these blood vessels and the thickness of the wall and all will be affected. Now let's stop here for a second to go over this picture. Here we have endometrium shown by this blue lining right here and this blue cell, this blue box. Do you see blood vessels right here and here, and how they go deep into the myometrium? Remember, this is the myometrium all the way to the perimetrium. Now, Nemish, when she has her menses, what's happening? You are shedding this top layer. Portions of this top layer are being shed. Okay? That's what's happening during menses. Now, when we have a fertilized egg ready to implant, it will burrow into this purple space and use these blood vessels to form a nice connection with. That connection is going to have two sides. One side of the connection is going to be the placental side, which is the baby's side. The other side of the network is going to be mom's side of the placenta. So the placenta will have two sides. One's the baby's side, one is the mommy's side. And they're all going to be within that purple area. Now, will the blood mix? No. Mommy's blood and the baby's blood will not mix. That is why some drugs will go across the placenta and affect the baby as well as the mom, and some drugs will not go across the placenta and will only affect the mom. In the immune system chapter, you learn that certain types of lymphocytes will actually go, certain types of immunoglobulins will actually go across the placenta. That's because those actually cross this barrier. So Nemish, is this almost like the blood-testes barrier and the blood-brain barrier? You bet. This is now the blood-placenta barrier. Okay, so the blood-placenta barrier is going to form here. Okay. So you're actually going to have sort of a airlock, and that's how I've always imagined it, an airlock between where the two airs never mix, or in this case, two bloods never mix. Okay, moving on. Okay, this one, just read through it. It's important, but read through it. Okay. Next line, next slide. Blood vessels are mentioned. Don't care too much about that. Okay. Really don't care too much about that. Okay, now you see the numbers here. So the menstrual cycle says lasts from 21 to 35 days. But I've said we're looking at the average female number being 28 days. So we're going to stick to that. Nemish, can those 28 days vary in the same individual? Yes. Sometimes longer, sometimes shorter. A whole lot of factors affect why it becomes shorter or longer. So our day 14 that we mentioned for ovulation can actually be altered. Now, down here we see menses. We're now talking about the uterine cycle or the menstrual cycle. We're no longer talking about the ovary. But what do you see down here? Ovary mentioned. Now, I already told you that when will menses occur? Day 21? No. When? I mentioned it'll occur when? Day 1, which will come after day 28 if we assume a 28-day cycle. So it'll be on day 29, which is really day 1, day 2, day 3, day 4, day 5. So those first 5 days of the next cycle is when menses will occur. Now at the same time, I mentioned to you that while menses is going on, the ovaries are actually starting a new follicular phase because of FSH. So yes, while she's having her menses, what will happen in the ovary? FSH will start a new egg to develop. All right, then we have secretory phase. During this phase, which is going to be after day 14, you're going to secrete. Now, what are we secreting? We're going to have the ovary, by way of the corpus luteum, to do what? Produce progesterone and estrogen. That is why it's called the secretory phase, because those two hormones are going to develop the endometrial lining. It's going to secrete the mucus to thicken and to improve the blood flow within the endometrial lining. So this slide, especially this portion down here, is important. Notice, uterine cycle responds to hormones from the ovarian cycle. So those two are related cycles. So what goes on in the ovary affects what goes on the endometrial lining. Next slide. This one I'll let you keep in mind. It's day 1 to day 5 typically, and this is what happens. Reduce blood flow, oxygen, and you disintegrate the functional zone, which is the lining of the endometrium, and pieces of the endometrium slough off. Next slide. Here's what happens. As a matter of fact, I just said it. So menses, degenerating tissues break away, they enter the uterine lumen and they go bye-bye. And you're pretty much tearing apart this. That's why it bleeds. Next, here's that zone, all up here. All this is torn apart and it bleeds through. Next slide. Menstruation lasts between 1 to 7 days. Again, I said 1 through 5 because that's the typical amount of time. Some people last longer, some people last shorter. Now, I will tell you that as you get older and you approach menopause, as you're approaching menopause, menstruation will become less and less. So for women who are watching this video, you're going to find that when you get closer to menopause, the number of days involved for menstruation and the number of heavier days versus lighter days will change. Okay? And you're losing some blood as part of the bleeding and tearing of the blood vessel connection on the endometrial lining. It's not going to kill you, but it is enough to make you faint for some people. Okay, prolific phase. Again, this slide is good, but I'd rather have you see it in picture form. This slide is good, but I'd rather have you see it in picture form. Okay. So again, you now see this zone developing. See how it says uterine glands? These glands are going to help develop and thicken the endometrial lining. So you could say that the endometrium is a glandular one, and it was mentioned earlier. It is a glandular thing. It's producing secretions. Next slide. This slide is important. Okay. So now let's go through this slide. So this is the secretory phase happening after ovulation. Look what it says. the endometrial glands will enlarge, increasing their rate of secretion. What is it doing? It's creating elongations and spirals. So why am I elongating and creating a spiral in that wall? Because it's the spiral for which the blood vessels will soak into. You're actually creating what in my mind is like a pool. You're creating a pool of blood in the thickened wall of the uterus. Okay? And when you shed off during menses, you're just shedding off that lining that contained the pool of blood. Okay? That's how I see it. Okay? So a good example of one would be a kid creating little tracks of sand at the beach. And then water fills in the sand tracks, and then he takes a little bulldozer or a shovel and wipes off the sand again. Very much like that. So you're creating a little funnel-shaped passageway that will have blood going through it. Okay? Good example. Begins at ovulation and lasts as long as the corpus luteum stays intact, which is how many days from ovulation to corpus luteum? 2 weeks. Now, to prove it, what does it say down here? Generally lasts 14 days. 2 weeks. Look what it says next. Ends as corpus luteum stops producing the hormones. So at the end of 14 days, which would be 28 days, you're going to have the corpus luteum go bye-bye and turn into corpus albicans that I mentioned before. Next slide. So here's what it looks like. See how all of these are now little tubes through which blood will flow through. Okay. So tubes through which the blood will flow through. Next slide. Okay. Now this slide is worth writing down. Okay, so this slide is worth writing down. You have menarche, that's when it starts, and then you have menopause, when it typically starts to slow down and end. Okay, so 45, 55. And in some countries, puberty starts early. Some countries, puberty starts later. Okay. So menarche and menopause. Next slide. Amenorrhea. Now this is something to keep in mind. Notice, physical or emotional stresses. Because if you affect your emotional state and affect the brain and affect the FSH, LH, GnRH and all that, that can affect amenorrhea. Then you have physical. If you're too exercising, if you over-exercise and you always do that, that can affect it too. You've heard of women athletes having problems with their period because of this. Next slide. Okay. Vaginal tract, highly distensible. I'll leave it at that. Okay. Don't really care too much about this. Again, I want to see this on a model. Okay, so on a model. Next slide. Okay. We've already talked about this. Don't care too much about it. The only part I might care about is this nerve down here, the pudendal nerve. That's the one that you saw in the male as well, and that's why I would say it's important. Okay, next slide. Okay, let's get back to where it was. Okay, here we go. Next slide. This one is worth writing down. You already know it's the inferior portion of the birth canal. Sperm go up it during sexual intercourse, and it's the passage and elimination of menstrual fluid. So that's sort of commonsensical, but it's worthwhile writing. Next slide. The vaginal tract. You know that the vaginal tract is always moistened. Okay, so there's always movement of fluid in that vaginal tract. Hymen. The slide is great. Just keep that in mind. So hymen. Next slide. Okay, so hymen. Okay, I don't really care for this one. Okay, this one, don't care too much about, but this one slide is worthwhile keeping in mind. Remember, the vaginal is thick and elastic, contains blood vessels, nerves, and lymph. Now, this is where I heard about some weird things where some people will put vodka dipped in you-know-what and put it in you-know-where. Okay? Now, vodka, being a strong alcohol, can be absorbed by way of the blood vessels in the vaginal tract. Okay. I just want to let you know that what some people are doing and how dangerous that can be. That can get you drunk real quick. Too much alcohol absorbed that way. Next, this is worthwhile making a note of. Okay. This is the picture. I do not care for this one. Okay. Next slide. Now, this slide is important. So this one I want to go line by line. The vaginal bacteria, a population of harmless, resistant bacteria. This bacteria is good bacteria. You do not want to mess with it. Supported by nutrients in the cervical mucus, creates an acidic environment and prevents growth of many pathogens. That is why the best natural way of dealing with problems is to let it go natural. You do not want to be messing with that vaginal tract. There are cases where if you have had too much intercourse back to back and the vaginal tract is not able to return back to its normal acidic environment with the right bacteria, that excessive having too much of a intercourse session back to back, not letting the vaginal tract come back to normal can cause yeast infections and things like that. So that's what this goes with. Okay. It is a good idea to get a vaginal smear done because it lets you know about things like how your cells are doing. So if you see strange cells, you might have a vaginal or an ovarian or some kind of cancer issue. So having a pap smear done, that's what this is all about. Next slide. Okay, I really don't care for this one. I don't care for this one too much. Just want to let you know that this is what allows for the lubrication of the opening. And of course, the clit and the clitoris is very, very similar to the penis, as mentioned here. The clitoris is analogous to the penis. Next. Okay. We have the Bartholin's gland. This is very, very similar to the Cowper's gland, but the Bartholin's gland is to lubricate the vaginal opening. So this is what lubricates the vaginal opening, very similar to Cowper's gland. Next slide. Don't care for that picture. I do care for this picture because this one does show up on the lab model. So lab model be able to work with this picture. Next slide. Okay. Don't care for that. Don't care for that. Don't care for that. Do not care for that either. Do not care for that. Okay, don't care for those. Okay. Now, this one is an important one. It's a summary of what we've talked about so far. So hormones are related to all that we've talked about. You know hormones are related to ovulation and hormones are related to the uterus and getting it ready. Next slide. So this one is a summary. Now, as with male, on this slide, we have our story about GnRH. It is going to be a copy pretty much of what we talked about in the male. Look what it says. GnRH relates to estrogen that increases the pulse and progesterone that decreases the pulse. Okay, so there is a stimulation here. Next slide. Okay. Don't really care for that. Don't really care for that because there's a chart eventually on this. Don't care too much about that. Don't care about that. Don't care about that. Don't care about that. Now I do care about this. So this is a summary chart that we've been talking about. So this is a summary chart to keep in mind. This one's important. You don't need to draw it, but you do need to understand how this plays a role. Next slide. Okay, this slide is important. Stimulates bone and muscle growth. That's very important in the female. So when menopause comes around and estrogen starts going bye-bye, now let's think about this. If estrogen is going bye-bye, it probably means that we're not developing the follicle within the ovary. Remember, who is producing the estrogen? The developing follicle in the ovary. Now, how does that relate to number one there about bone and muscle growth? Well, estrogen plays a role in bone retention. Hormone estrogen plays a role in bone retention. When estrogen goes bye-bye, calcium comes out of your bone. It also plays a role with muscle growth. Number two, maintains female secondary such as body hair and adipose tissue. So if females have more testosterone, they start getting all that hairy face. And the way their body adipose tissue deposits right around their thighs and their buttocks, that's all because of estrogen. Number three, affects the central nervous system, where estrogens increase the sexual drive. That's why after menopause, sexual drive goes bye-bye for both men and women. Number four, we've already looked at that. They play a role in the reproductive glands. And the last one, initiates repair and growth of endometrium. So when the ovary no longer produces the follicle, which again no longer produces estrogen, you're not likely to maintain and develop the endometrium. So even the endometrium is going to go bye-bye. It's not going to be nourished well. That makes it very difficult to get pregnant later. Okay, next. So this slide is important. Next slide. This slide and the other half is found on page 1066. Now, does it look anything similar to what we've had in the male counterpart? You bet. So it's very, very similar to that. So much of what I've said can be summed up by page 1066, and I'm moving now to page 1067. So 1066 and 1067. Now, at the very beginning of the female part, I indicated that what happens with the hormones is going to be related to what happens in the ovary as well as what's happening in the ovary itself. So now we have the ovary, the follicle, and there's a piece missing which is on the next slide, which is the endometrium. So if you actually check that, you'll see it. Now, definitely study page 1066, 1067. Now, what is this basal body temperature down here? Well, one of the ways you can sort of predict ovulation is by the rise in the basal body temperature. Now, is it accurate? To some degree. Now, if you have a fever that offsets it all, then that's not going to help. But typically, here's what they do. A couple that's having trouble getting pregnant because they can't time it right, what they'll do is they'll use a calendar. And on every single day, the female will take a body temperature by way of mouth. Okay? So they'll take a normal oral body temperature, and they'll record it every day and take it at the same time of the day every day. When they see a rise in the body temperature about that time, and you don't know when that day 14 will be, it doesn't have to be on the day 14 of the month. Remember, cycles of different women are on different days. So their cycle may start in the middle of the month, the front of the month, the end of the month, we don't know. So at some day, which corresponds typically to their day 14, they're going to have a rise in body temperature. And when that body temperature rises, it's going to stay elevated in comparison to prior to ovulation, which is what you see on the screen right now in front of you. So the rise in body temperature and the maintenance of the body temperature for several days will indicate that you're in that new phase after ovulation. So it lets you have an idea of when to have intercourse. Typically, it is 1 day to 2 days after ovulation that you want to have it done. So 1 day before or 2 days after. So 1 day before, 2 days after. That's typically the window. 1 day before, 2 days after. And if you saw that with me, 1 day before, 2 days after, will relate to the roughly 3 days that the sperm would be relatively viable. Okay? So that's what this relates to. So now, here's day 1 to day 5. And if we round that off from 1 to 5, you see that we have our menses here. So menses is happening day 1 to day 5 roughly. And if I go to the previous slide, that's corresponding to the development of the follicle, which corresponds to the rise in estrogen. So as the follicle will develop, so will the rise in estrogen. Now, I have not mentioned this part yet, but notice here, the rise in estrogen and the development of the follicle corresponds to FSH. Then around day 14, we have a surge in LH. It's just the right day before. So the day before, you're going to have a rise in LH. That rise in LH the day before is going to stimulate the ovulation process. Also, we mentioned corpus luteum, which will take place after day 14. And notice it'll go from day 14 very, very close to day 28. And you can see that progesterone is at the highest right when we have our corpus luteum. So it's the highest right when we have our corpus luteum. Okay. By the time we have corpus albicans, we see our menses starting to form. And you can see that on the next slide. So as I have progesterone high, I'm going to have my endometrial lining well nourished. And notice, all I'm doing is making a little valley, a spiral tube for which blood can pool through. Okay, that's all it's doing. Some people use the word lacuna from 2401. Basically, to me, it's the same thing. Okay, so make sure you're able to relate this picture with this picture. Okay, next slide. Okay, this slide is a summary of what we've said so far. Okay. Next slide. Okay. This continues our story of what we've said so far. Next slide. Okay. This is also on the chart that we've just mentioned. Look what it says here. Ovulation occurs 36 to 38 hours after the LH surge. Okay? That's about a day before ovulation. Okay? Some people say a day and a half, maybe 2 at the most. Okay? So a day and a half after, it says 9 hours, so about a day and a half after, or about a day after the surge. Okay. Next slide. Okay. We've already talked about this as well. Okay. We've already talked about this as well. Now, here's the magic. Unless pregnancy occurs, the corpus luteum begins to degenerate. Now, that line is important for the following and last chapter, which is chapter 29 on development. It says that if Friday night, Saturday night doesn't happen, then the corpus luteum will do what? Degenerate. And you're going to get your menses. However, if the egg does get fertilized in the isthmus of the fallopian tube, and the egg, the fertilized egg, makes it to the endometrial lining and starts to burrow its way in the endometrial lining, using the blood that I mentioned as a way of nourishing and setting up the placenta will tell the corpus luteum to continue beyond its normal 2 weeks. Let me say that again. So if we do have fertilization, we do have implantation, and the fertilized egg is now ready and able to make the placenta, then yes, the corpus luteum will be told by the placenta to continue beyond the 2 weeks or the 14 days. And that's also one of the ways you know that you are even pregnant. So when the placenta produces a hormone that we typically call our birth, our pregnancy detector, the human HCG. So when you see the HCG in the blood or in the urine stream, and you know it's enough to come from the placenta, then you know that the corpus luteum will be maintained beyond the 2 weeks. Otherwise, it will degenerate. Okay? And again, ovarian cycle starts all over because FSH is told to stimulate. Okay. So again, keeping page 1066, positive feedback, negative feedback thing in mind, along with page 1067. All right, we've also talked about this. When progesterone and estrogen go bye-bye, because the corpus luteum goes bye-bye, what will happen? Menses. And it's going to shed for several days, usually 1 to 5. Okay? Until the rising estrogen stimulates regeneration. So when the next cycle starts all over again after day 5. So after day 5, because of what? The developing follicle producing more estrogen, you're going to develop the endometrial lining all over again. So as we get to menopause, you're going to have less and less likelihood of developing the endometrial lining. Next slide. We've already talked about that. We've already talked about the body's basal temperature. Okay, so day after ovulation, the temperature rises, and I've already mentioned this. It's not the best way, but it does give you some idea. Next slide. Okay. Coitus, or copulation. Okay. Introducing semen into the female tract. Now, wait a second, Nemish. How come it says autonomic here? That's because even things that work down there are controlled by up there in the brain. It's an autonomic function. Okay. Now, having said that, what kind of words do you see on this slide? So where am I in the chapter? I moved over to page 1068. What's mentioned here? Words like arousal, erection, sympathetic, parasympathetic. So it is what? It says leads to increased parasympathetic. So just like your digestion, just like your urinary, the on mode is parasympathetic when it comes to sexual function. So the on mode is parasympathetic for urinary, digestion, and sexual function. Now, let me back up for a second. You see the words starting with an E: erection, emission, ejaculation. Make sure you know what those terms are. So, erection, emission, ejaculation. Next slide. Okay. We've seen a lot of these terms already. So this is now under emission. So this is under emission. Remember the 3 E's: emission, ejaculation, and erection. So this is emission. Okay, so make a note of that. Here's ejaculation. Okay. Make a note of that. Okay, and now we're on impotence. So the inability to achieve or maintain an erection, often caused by either a physical problem or a psychological problem. Okay. Next slide. Okay. Female. Again, what do you see? Parasympathetic. So are you seeing a trend here? Anytime it's a natural process where you're not dealing with stress, a natural process, it tends to be parasympathetic. Next slide. Okay. So again, not too concerned with this. Okay. This one is important to me. So be aware of these diseases. It is important for test purposes. So definitely know a summary about these diseases. Okay, so a summary about these diseases. Next slide. Okay, now we come to aging. Like every chapter pretty much ends with aging. Men and women do have a form of menopause. So men and women do have a form of menopause. For typical ages, we're talking about 45 to 55. It is a time that ovulation and menstruation cease. And of course, by now you should understand why, because these are interrelated actions. Circulating concentrations of estrogen and progesterone decline, and GnRH, FSH, LH rises sharply because you don't have the negative feedback influence. So you don't have the negative feedback influence. Next slide. Perimenopause, just to have an idea of what that is. Okay. Here's what happens when you have estrogen levels drop. I did mention a couple of them already. Reduction in the uterus, so the uterus will actually shrink in size. Okay. Breast size will go, because again it's hormonally related. The thinning of the urethral and vaginal, that makes sexual intercourse very painful because they're thinning out their vaginal lining, and they're also cutting down on the lubrication, so the lubrication goes bye-bye, so intercourse becomes more painful. And then of course we have osteoporosis because of the estrogen going bye-bye. Next slide. Men have it too, but notice the men's age is a little bit higher. It comes because of decrease in testosterone. Now, that causes FSH and LH to rise. Remember, you don't have the feedback anymore. Sperm production will still continue, and sexual activity will gradually decrease because of declining testosterone levels. So sex drive will drop with the testosterone level. Next slide. Now, for males, sperm count must be adequate. Semen must have the correct pH and nutrients. Now let's backtrack for this line right here. If you have your prostate going bye-bye, which is what tends to happen when you age, what's going to happen to your semen? It's not going to have the right nutrients. So the semen quality will drop. Okay? Erection and ejaculation must be proper. That means your hormones must be right. Your nervous system must operate correctly. Next slide. For females, you must have the right coordination between the ovarian cycle and the uterine cycle. So what's happening in the ovaries must properly match up with what's happening to the endometrial lining. Of course, the egg has to get to the right place. You have to have the right environment. Survival of sperm and the movement of sperm is important. And you must have it fertilize right at the isthmus of the fallopian tube. Now, I told you earlier that's really more of a female issue, 80 to 20 kind of thing. That's because there are more issues that can happen in the female, such as, 1, the vaginal tract has the wrong pH. It's killing off the sperm even before it gets going. Second, the sperm don't make it there because the cervix is having a very thick mucus. So the sperm never make it past the mucus and she can't get pregnant. Okay, so that's a problem. Then, let's say the sperm make it up there and they get to the fallopian tube, but the fallopian tube is blocked by scarring. Because of some illness or injury or whatever, there's a scarred fallopian tube and the sperm can't make it there. All right, let's say it's fertilized, but the egg doesn't get a chance to implant because the endometrial lining is somehow not ready for implantation. So there's many, many possible things that can go wrong, and most often it's the female. Okay, next slide. Okay, these are the hormones that we've spoken of. Of course, by now you should know where they come from. And notice here the author has done a good job of breaking it down to male, female, male, female. What I would do to best understand this is to actually separate it. Create your own chart. You may have already done this when we were on chapter 18. But if you have not done it, for understanding this chapter, it is best to separate them. So separate them with what happens in the male versus what happens in the female. So I would definitely separate out these charts. Next slide. And in order for the reproduction chapter to work, all this has to work. You've got to have a right blood supply to the penis and to the endometrium. So the penis and the endometrium have a good blood supply. You saw parasympathetic, sympathetic playing a role. It's got to be right. The endocrine system has to be right. And of course, the reproductive system has to be right. Okay? That takes care of chapter 28.